Hello guys, this is Naman Trivedi. We'll start with the imbibition topic. So we are into the fourth lecture of the transport in plants chapter. So I hope you are following me, you are for reading the lectures, you are going through the lectures and you are reading well. And keep on preparing appropriately. Okay? And kindly keep responding in the comments on my YouTube video so I can solve your doubts and I can help you out. So today we'll start with the imbibition and then the long distance transport of water. That is what we'll take in this lecture. Okay, so we'll just start with the imbibition. Hold on for a second. Yeah, see ya. Yeah, excuse me for that. Yeah, so we'll start with the topic of the imbibition. Okay, so imbibition is as such very easy, but they have asked questions from it, and it is very easy to be understand imbibition. I'll just give a simple example. If you take a glass of water, okay, and if you fill it with the couple of dried grapes, okay, or what we commonly call as kismis, okay, then the next morning you will see that all the kismis or whatever the dried grapes, they are all bulged up, or they are all basically swelled. So the swelling happens because the water has entered inside it. So imbibition can be defined as a special type of diffusion when the water is absorbed by solids or colloid. So remember, imbibition is defined as the absorption of water by solids. Okay, it is defined as the absorption of water by solids. Okay, and that's why this water absorption will increase the volume. So keep that in mind. Now the classical example which your textbook has mentioned is the imbibition of water by seeds and dry wood. You might have also observed that even at our homes during the monsoon or during the rainy season, the wooden doors will expand a bit, and that's why you have to push it a bit to close them or to open them. That is because it absorbs moisture, that is mainly water, and it swells. So this knowledge has also been obtained by the prehistoric men, and they used to use this swelling. They used to just a moment, excuse me. They used to use this capacity or this swelling to mainly split the rocks and boulders. Okay. Then the next example is commonly the seedlings which come out of the soil. Okay, or when the germination happens. That germination and seedlings coming out of the soil can be also be termed as the process resulting due to imbibition. Okay, now next is imbibition is also a diffusion. As I told you, the water movement happens from same along the concentration gradient. So imbibition is a movement which happens along the concentration gradient, remember that. Okay, so basically the seeds and all this material will absorb the water easily. So water potential gradient will be formed from where there is high psi W to where there is low psi W. Okay, and this will lead that liquid is imbibed, and that is what we call as imbibition. So for any substance to imbibe the liquid, there must be affinity between the absorbent and the liquid. So there must be some sort of attraction or affinity between adsorbent and the liquid is prerequisite. Without this, water cannot diffuse and imbibition cannot happen. Without this, water cannot diffuse and imbibition cannot happen. So just a moment. Yeah, so for imbibition you need to know it is nothing but it denotes absorption of water by what we call as solids. Absorption of water by what we call as solids. Second thing is imbibition is mainly based on absorption by solids or colloids. Okay, then third thing is imbibition. The common example is the seedlings emerging out okay, then wood absorbing water, okay. Then I just give you the examples of these grapes, dried grapes which you keep in water overnight, and in the morning they will be swelled, okay. And also the last thing is, it is the movement of water will always from high water potential to low water potential, from high water potential to low water potential. Other thing is, there will, it is always along the concentration gradient, it is always along the concentration gradient, remember that. Also the gradient, okay, is from high to low, so gradient is again from high concentration, sorry, the gradient is again from high concentration to low concentration, remember that. Also imbibition, for imbibition to happen there must be some sort of affinity between the absorbent or adsorbent which will absorb the water and the substance. Imbibition is simply based on what we call as diffusion. It's simply based on what we call as diffusion. So that was how I can sum up the entire part of the imbibition. Okay, so it's easy, but they do have asked questions from the definition part, so just go through this part. Okay, now next is we'll discuss the long distance transport of water. We'll discuss the long distance transport of water. Okay, so kindly pay your attention. So now they are mentioning in this paragraph that you in the lower standard you might have done the experiment that you place the twig having white flowers in a colored water, and after a while you might observe that the colors of the flowers has turned into the in the water color you have put it. So let's say if you put the flowers in the blue colored water, the flowers which were white will become again blue in color. So you when you cut the end of the twig after a while, okay, you'll see that there is a region inside which there will be movement of the water, okay. So it is basically the movement of water which will go from vascular bundles, and more specifically this movement happens through the xylem, okay. More specifically, the movement of the water or colored water has been absorbed by the twig with a which has the white colored flower, and the route taken is mainly through the vascular bundle, through the vascular bundle as well as mainly in that again through the xylem. As we know, xylem is the, as we know xylem is the vessel, okay. Xylem is the vessel which can mainly, it is responsible for H2O movement, it is responsible for H2O movement. So how this long transport happens, because the water was colored, it has entered inside the plant, and then the plant flower color has changed, okay. So here, as you know, plant bodies are very big, I mean, so plants can be up to like 80 meters in height, for example, okay. So for this distance to travel, there is some mechanism through which what we call as the long distance transport of water. So it cannot be done by diffusion. Long distance can never be only done by diffusion, because diffusion is a slow process. So as we discussed earlier also in the first lecture of this chapter, that diffusion accounts for only short distance movement. Diffusion accounts for only short distance movements. So for long distance, okay, they've given this over here, they've given this example that for movement of a molecule which is of a typical plant cell, okay, like which is 50 micrometer in size, it takes approximately 2.5 seconds, okay. It takes approximately 2.5 seconds, okay. Now at this rate, this is the rate of diffusion, okay. If everything will be moved, then it will take possibly millions of years, okay, okay, of to travel distance of a 1 meter, because here as you can correlate that for movement like 50 micrometer, it is taking 2.5 seconds. For 50 micrometer, it is taking 2.5 seconds. Now if you want to move 1 meter, how much it will be? You can cross multiply it, okay. Then you will get it, it will probably take thousands of years. It will take very long time. It will take very long time. So that is what they are trying to explain in this part, okay. So diffusion is not possible because it is very slow process. So now how the movement will happen, because we have seen there are very huge trees like gymnosperms, you remember sequoia trees, okay, which are the tallest trees on the earth. Then in the large and complex organisms, as we moved, like in plants, for this movement there must be some sort of mechanism. So that's why for long distance movement there is something termed as mass or bulk flow system. There is something termed as mass flow or bulk flow system. There is something termed as mass flow or bulk flow system. Okay, now uh, it mentions that water and minerals are usually moved, or food is generally moved by mass flow or the bulk flow system. Okay, so mass flow movement is defined as substances which move in bulk or en masse, substances which move in bulk or en masse from one point to another. So they are using a term called en masse. They are using a term called en masse. They are using a term called en masse. Okay, en masse means mass or bulk flow. Okay, en masse means in some quantity. It's a French term which means if some substance in some bulk amount, if some substance in some bulk amount has been moved from one place to another, we call it as a en masse movement. Okay, and this is this happens because of the pressure difference. This happens because of the pressure difference between 2 points. So en masse movement happens because of the pressure difference between 2 points. That is what they are trying to explain. Okay, so how this pressure difference will be created? Okay, so they've given the example of a flowing river. So they've given the example of a flowing river. So if you talk about the flowing river, okay, if you talk about the flowing river, then in the flowing river, because of the force, there is bulk flow can be seen. So whatever comes along, the river will take it, be it the clay material, be it the stones, be it anything. So it is again an example of what we call as en masse movement. That again an example of what we call as en masse movement. Okay, so here the movement is independently of the concentration gradient. The movement is independently of the concentration gradient. Now here they have explained two things. Here they have explained two things, that is positive hydrostatic pressure gradient and negative hydrostatic pressure gradient. So remember that to achieve the bulk flow, remember that to achieve the bulk flow, to achieve the bulk flow, there are two mechanisms. There are two mechanisms. Okay, what are they? So the first one is it is achieved by either positive hydrostatic pressure gradient, or it can be achieved by negative hydrostatic pressure gradient, or it can be achieved by negative hydrostatic pressure gradient, as they have mentioned over here in this last. I'm just highlighting it, this part. Okay, over here they have mentioned. Now basically the mass flow is achieved by what we call as the positive hydrostatic pressure gradient or negative hydrostatic pressure gradient. Okay, now understand the example of the positive hydrostatic pressure gradient can be, they have given the example of garden hose. They have given the example of garden hose. Okay, so I'll just give you the example. I'll explain about the garden hose. So garden hose, there is one source from where the water is coming. Okay, and then it will go towards some particular direction. So let's say this is where we have the hose which will the supply is it is taking, and then the water is being passed on to the plants. So this is an example of positive hydrostatic pressure gradient. Why? Because water is coming from the place where it is more in amount and it is transferring to the negative, or the where there was no water. So such a gradient from positive towards negative is termed as positive hydrostatic pressure gradient. Positive hydrostatic pressure gradient. Second such example is of negative hydrostatic pressure gradient. So if you talk about the sucking by straw, okay, so let's say over here you are drinking, this is the coconut water you are drinking, and this is the coconut, so this is what we have, straw. Okay, so in this case it has been sucked, the force is applied from this direction, you can see the arrow, by your mouth you are sucking it. Okay, so this creates a negative hydrostatic gradient, this creates a negative hydrostatic pressure. So remember the example of positive and negative hydrostatic. Positive hydrostatic example is of the garden hose, and negative one is the sucking of by the straw, which we all have done okay, while consuming different liquids. Okay, so remember these both examples, okay, about the positive and negative, and these are the mechanisms responsible for what we call as bulk flow. So basically the bulk flow can be defined as the movement of substances, okay, the movement of substances which occurs through vascular tissues of plants, and that is what forms the basis of translocation. So the bulk movement which happens through the vascular tissues of plants is what we call as the translocation. Okay, the bulk movement which happens through the conducting or vascular tissues is what we call as translocation. Translocation is important for question. Okay, they asked this definition. So remember the definition of translocation. Okay. So as we know, we have plants have xylem and phloem, the specialized vascular tissues. Okay, for translocation of water, minerals, food material, nitrogen, hormones, everything. Okay, so depending upon the type of tissue, they will transfer different things. So the next part is mainly about how do plants absorb water. So in the plants, mainly water is absorbed, okay, through the soil by the roots, as you all know. Okay, so in the plants we all know that the water is mainly absorbed by the soil. Just a moment. Yeah, so roots are responsible for absorption of water. Okay, then if they talk about the minerals, then minerals are mainly absorbed by root hairs, okay, which is on the root. So over here they have mentioned roles of two things. Okay, we should know the role of two things, that is, first is about the role of roots, okay, role of roots, and then role of root hairs. So root hair will increase surface area, it also helps in absorption, you just go through this paragraph, you'll come to know. Water absorbed along with minerals by the root. As I told you, water cannot always be in pure form, and soil is the richest reservoir of minerals, so the root hairs will absorb it mainly by diffusion. Okay, after absorption by the root hairs, water can move by two distinct pathways. After absorption by the root hairs, water can move by two distinct pathways, that is, one is apoplast and second is what we call as symplast. One is apoplast and second is what we call as symplast. Okay, so we'll discuss and we'll start with the apoplast pathway. Okay. Now here if you look at this diagram, okay, they mentioned the water movement mainly. Okay, so you can see that these are all cells, which they are trying to show you. Okay, now as you can see their cell wall, this part, it is continuous except the place where there is Kasparian strip. So at this region, So I'll just change the color so you can understand. At this region you can see there is something termed as Kasparian Strip. So at this region there is something termed as Kasparian Strip. So this is what the Kasparian Strip is. So till there they have this cell wall thing and the movement can happen. Now this Apoplast movement, okay, will happen till the intercellular space and wall of the cell. Okay, so it doesn't cross the cell membrane. So as you can see the direction over here, the water which has been absorbed by this route, okay, will always go from this cell wall part. You can see this will always go from the cell wall part. Okay, so in this direction there is no issue, it can easily move. But the problem will be encountered at the Kasparian strips. The problem will be encountered at the Kasparian strips. Remember that we are discussing apoplast. Okay, now this movement is mainly based on the gradient. This movement is based on the gradient. So it is, remember, from high concentration to low concentration. It is, remember, from high concentration to low concentration. Okay, so now just I'm removing it just so that I can write something further. Wait for a moment. So there is no barrier for the water movement in the mass flow till it reaches the Kasparian strips. Okay, now these mainly Kasparian strips are encountered at the endodermis. So outermost layer, if you remember the plant anatomy chapter, the outermost epidermis, then we have the cortical part, and then we have the endodermis. So this apoplast pathway is followed through cortex, epidermis, cortex, and the problem only occurs at the endodermis. EP stands for epidermis, EN stands for endodermis. So here it will encounter the Kasparian strips. So till this route, okay, till this route it will undergo through the apoplast. Till this route, till the Kasparian strips are encountered, there will be apoplast pathway which will be through the cell walls. As you can see, it is only going outside, it will not enter in the cell. It will only, the movement is only through these cell walls. The absorbed water will only move through the cell walls, remember that. So there is no crossing of the membrane. Okay, now try to understand that apoplast does not provide any barrier to water movement, and water movement is through mass flow. So in the apoplast, water movement is through the mass flow, that you should know. Okay, so this pathway which we are discussing, apoplast, this pathway through apoplast is always through what we call as mass flow movement. Okay, now as the water keeps on evaporating from the intercellular spaces in the atmosphere, it will generate a tension, and hence the mass flow will occur because of adhesive and cohesive properties of the water. Because of adhesive and cohesive properties of the water. So I'll explain you how it is. So let's say from this place the water is lost. From this place the water is lost. There is evaporation. So this is where water has decreased. Okay, so from where this plant cell will take water, so this will take water, this will take sorry, just a moment. Let us assume that from this place water has been lost by evaporation, so it is decreased, so I am putting minus sign. So from where it will take water, it will take water from the nearby cell where there was more. So now this will again lose the water, this will again lose the water, so it will again become water decreases. So where this will take water from, again the next cell, next cell. So in this way the entire channel is formed. So in this way the entire channel is formed, that the cell will take it from the next part, next cell, and it will continue. Now this entire column channel will be formed, like let's say if you take a single plant, I'm just drawing a schematic diagram of the plant. So this entire chain, okay, will form from this will to this place. Okay, so and then it will expand till the roots, it will expand till the, because here the water is lost, by evaporation. So it will expand till the roots, and then the plants will basically form this entire column. Okay, and the roots will keep on absorbing water, and that water will reach to each and every part of the plant from the roots, and it is lost mainly by what we call as evaporation. It is lost by what we call as evaporation. This is what it has been explained in this part. The tension will develop, okay, because of evaporation, and that's why the water will keep on absorbing. Okay, the Symplast movement will happen through the protoplast, that is, it is through this Plasmodesmata. Okay, as you remember. So now try to understand. Okay, I'll just again explain. So what happens in Apoplast, what we have discussed till now, that whatever water has been absorbed by the root hairs, okay, will go to the epidermis. From epidermis it will go to where? From epidermis it will go to the cortex cells or cortical cells. From cortex it will go to the endodermis. Okay, so the simple route is epidermis, cortex, endodermis, and then it will encounter the Kasparian strips. Just a moment, let me have some water. Okay, yeah. So till that part the movement has happened through the apoplast movement, and most of the water mainly moves by the apoplast only in the roots. Okay, the problem happens after the Kasparian strips. Now second movement which they are trying to explain is the symplastic movement. Okay, so Symplast movement mainly happens because of, just a moment, yeah, so it is by the system of interconnected protoplasts. So I hope you know what are protoplasts. So protoplasts are the cells without cell wall. So it is what we are talking about right now, about the cell membrane, not about the cell wall. Apoplast was through the cell wall. Now they have also explained one more thing, that is, there is something called the cells are in contact with Plasmodesmata. I'll just explain to you what is Plasmodesmata. Just hold on a moment. So let's assume, if you remember the cell chapter, I also discussed this thing, that we have, this is the plant cell, okay, and this is the another plant cell. So these plant cells are in contact with one another through these cytoplasmic strands, which will traverse through the cell wall, because this part, as you see, is a cell wall, and the cytoplasmic strands, the blue colored part, I am again highlighting them. See, you can see the contact is there between this first cell and the second cell. Okay, you can see the contact there between the first and the second cell through the cytoplasmic strands, and this is what, and these strands are what we call them as the plasmodesmata. We call these cells as mainly plasmodesmata. So plasmodesmata is a connection of cytoplasmic strands between two plant cells. So in the Symplast, water will travel through the cytoplasm, and the intercellular movement, that is movement inside the cells, will be through plasmodesmata. As water enters inside the cell, cell membrane, the movement is slow, and it is also as per the potential gradient. It can also be aided by cytoplasmic streaming, that is, inside the cell cytoplasm will, circular motion will be there, and it will move the water inside that. Okay, as you can see in the hydrolith experiment, if you have done it in your practicals. Okay, the chloroplast movement you can see easily in that. So the movement mainly happens in this part in the Symplast is through the Plasmodesmata. So I will just again explain the diagram. So if you can see this part, the movement happening inside the cell, this entire part, okay, that is what we call as through the cell membrane. And this red line is basically representing what we call as the Symplast. The red line is representing Symplast. So in the Symplast, just a moment. So in the Symplast, the movement is inside the cell. First and foremost, it is through what we call as protoplast of the cell. So it is from the cell membranes of cells. So what cells will participate? Same. The cells will be mainly epidermis, okay, then we have cortex, then we have endodermis till it will go to the pericycle, and it will basically enter in cell, okay. So the process of Symplast is slow, okay. It is also as per the concentration gradient. Inside the cell there is cytoplasmic streaming or movement of the cytoplasm. Inside the cell there is cytoplasmic streaming, okay. You might have done the hydraulic experiment in which you can see chloroplast movement, so that is the circular movement of cytoplasm, or what we call a cyclosis of cytoplasm will happen. That is what we call as this Symplast pathway. Once again I am explaining, okay, and then I'll end the lecture, that Symplast movement will be through this route inside the cell, this red line I am showing, whereas Apoplast movement will be through this part, so through this cell wall part, as you can see the blue line. It will occur till the Kasparian strips are encountered. So blue one is the, as you can see, the blue one is the Apoplast and the red one is the Symplast. Remember that. So it is all, we are still in the roots, okay. Now as we know, the resistance is encountered at the endodermis because there is Kasparian strips which is made up of suberin, as there is Kasparian strips which is made up of, remember that this is made up of suberin, okay. So over from that region water molecules cannot penetrate the layer, okay, because the walls have suberin deposition. So then it will go through this Symplast. So whatever apoplast movement which will occur till endodermis, the next phase, the movement at the Kasparian strips or after endodermis will be through the Symplast, because of this deposition of suberin, which is a sort of polysaccharide, it will not allow the movement of the water through the cell wall, okay, and then it will move through the Symplast, and ultimately it will reach to the xylem. So after that it will basically through the Symplast and reach to the xylem, okay, and then it will reach to the vascular cylinder or the vascular part where it will enter, okay. In the xylem water will move again from cell to cell. Okay, or xylem vessels or tracheids will play the role, okay. Non-living parts will help in that, okay, and that is what the route can be seen. As you can observe over here that this is the symplastic path, okay, going inside the cell and then reaching over here, this pink line I am showing, okay. If you talk about the apoplastic part, then you can see this are from the outside the cells, so as you can, I'll again explain once again. Through the apoplastic path the movement is from the cell wall and it will only happen till endodermis. As you can see, it will only happen till the endodermis because at that region you will be encountering Kasparian strips, whereas symplastic will be up from the root hair. It will be inside the cell through the protoplast. It is slower, and then it will reach to the xylem. So there is the most important path over here, that is the symplast, because without the symplast, water cannot reach to the xylem. So once again I will sum it up. Okay, apoplast is the movement. Okay, apoplast is the movement in which water will enter to the epidermis, to cortex, to endodermis, and then just a moment, yeah, and then will reach to the Kasparian strip. So the further movement is not possible. So after Kasparian strip, the movement is the symplast. After Kasparian strip, the movement is the symplast. Now Kasparian strips are made up of what we call as suberin. Kasparian strip are made up of suberin, which will not allow water to penetrate, which is not permeable to water. So then the movement is supposed to happen through what we call as the Symplast pathway. Now Symplast, basically, if we'll discuss about the Symplast, just a moment, I will just write over here so it will be easier for you to understand. So Symplast movement, if we talk about the Symplast, is mainly through the protoplast, that is, it is through the cell membrane. Okay, so it is inside the cell. Endodermis we have epidermis, then we have cortex, then endodermis, and then it will reach to the xylem part. Okay, then it will reach to the xylem part. So as you can observe over here, Symplast is through the cell. Okay, whereas epiplast was through the cell wall. Okay, now as the Symplast is mainly the water will mainly travel through cytoplasm. There is also a role of cytoplasmic streaming in that. Okay, and then to reach to the just a moment, yeah, and then to reach to the, let's say, endodermis, to the xylem, which path is followed? Again, it is through the Symplast. Okay, till endodermis it can follow both, that is, it can follow apoplast or Symplast. Okay, Symplast is also aided by cytoplasmic streaming, or what we call as cyclosis. So read it from the textbook. It is very much important. Apoplast, Symplast. If you have any doubts, you can ask me. Okay, so I'll just like to sum up my lecture. From today's lecture, what is important is you should know about the definition of translocation. You should know about definition of imbibition. You should know positive hydrostatic pressure. You should know negative hydrostatic pressure. Okay, then next you should know about the two pathways. Okay, or you should know the bulk flow or en masse movement, or what we call as bulk flow movement. Now this movement or water mineral absorption will be mainly by two routes, that is apoplast and Symplast. Okay, so apoplast is through what we call as it is through the cell walls. Okay, this is through the protoplast. You should also know about Plasmodesmata. Okay, inside the cell this movement will happen. So remember these things from today's lecture. If you have any doubts, you can ask me. Okay, that's all. Naman Trivedi here, signing off. Take care, stay safe, and keep mentioning in the comments about how you feel about these lectures. Thank you guys.